Hello and welcome back to Biology for Beginners. As always, I'm your host, Morgan. In the last couple of videos, we've been talking about cell division all the way from the basics of binary fission all the way up into the thing that makes sexual reproduction possible, known as meiosis. Well, today we're going to be discussing what happens when cell division goes wrong and how cell division can go wrong. As always, we are taking a couple of moments here to pop up those terms and definitions slides so that you can pause and grab the vocabulary we will be using in this video. But also a quick reminder that you can pop on over to my Patreon. The link is down in the description below, and grab my entire slideshow as a PDF for absolutely free. However, all patrons do get the Bio for Beginner videos and all of the accompanying study material that I put together at least a day early. There are also other videos that will go up at least a day early on the Patreon. However, we are still kind of working on that right now. But without further ado, let's talk about when cell division goes wrong. The first thing that we need to be aware of when we're talking about what can go wrong during cell division is a thing called a nondisjunction. Now, a nondisjunction is basically an accident during meiosis where the result is at least one gamete ends up with 2 copies of one chromosome and another gamete ends up with no copies of that chromosome. And this can occur during either meiosis 1 or meiosis 2, resulting from either homologous chromosomes not separating properly or sister chromatids not separating properly during either anaphase 1 or anaphase 2. A quick little aside is that this can occur during mitosis. However, it's not going to affect the next generation because mitosis produces somatic cells and somatic cells are not passed on to the next generation. Only the genetic information held in gametes is going to be passed on to the next generation. So we're going to focus on nondisjunctions during meiosis. So if the nondisjunction occurs during meiosis 1, what we're going to see, as we can see in this diagram here, is that these homologous chromosomes are going to fail to separate during the anaphase. And if you look at this first box in this diagram here, you'll see the smaller homologous chromosomes on top separate just like normal and go to opposite sides of the cell. But the larger chromosome on the bottom is being pulled entirely to that one side. So the 2 chromosomes did not separate. Both homologous chromosomes end up in one half of this separating cell for the meiosis 2 process that is about to occur. And then if this nondisjunction does occur during meiosis 1, usually meiosis 2 is going to proceed as normal, and the result is going to end up being 2 gametes with n+1 chromosomes, so 1 extra chromosome. Because remember, gametes are haploid, meaning gametes are n, so they have n chromosomes. And your usual somatic cells just bopping around are going to be the 2n diploid. So that's what all this n stuff means. And so the 2 gametes have n+1, signifying they have 1 extra chromosome. And then you have 2 gametes with n-1, so they have 1 less chromosome than what they're supposed to have. Now, this can be directly compared to a nondisjunction happening during meiosis 2. If this nondisjunction does happen during meiosis 2, it's going to happen after a meiosis 1 that proceeded as normal. So those 2 homologous chromosomes, in this case, both of them separated just fine. The little one on top, the big one on bottom, they both separated as they were supposed to. But at this second anaphase, during anaphase 2, in at least one of those daughter cells that was produced, you'll notice that that large chromosome on the bottom, the 2 sister chromatids, are not being pulled apart like they're supposed to be. They're being both pulled to one side of that cell. And the result of this is that you get 1 gamete with an n+1 chromosome count, 1 gamete with an n-1 chromosome count, and then 2 normal gametes. It is entirely possible for the nondisjunction to happen in both cells, but it's very unlikely and very rare. So in general, the result of the nondisjunction during meiosis 2 is going to be the one gamete with n+1, one gamete with n-1, and two normal gametes. Now, again, if you just look at both of the diagrams right next to each other, you can really see the differences. In one case, you get that nondisjunction during meiosis 1, the homologous chromosomes don't pull apart, and you end up with more gametes with the abnormal chromosome count. And then in the other case, you end up with only the two gametes with the abnormal chromosome count because the nondisjunction happened during meiosis 2. And again, nondisjunctions can occur during mitosis, but we're not really going to talk about that at all because that occurs in somatic cells, and somatic cells don't get passed on to the next generation. So how can you tell if everything is exactly as it should be in the genome? How can you tell if there's the correct amount of chromosomes in a particular cell or organism? Well, what you can do is you can look at the karyotype. And a karyotype is basically the tool that scientists use to actually visualize those chromosomes. They go through a process of centrifuging and staining the chromosomes, and then you end up with what we see here on the slide, which is a bunch of stained and ordered chromosomes that you can actually look at and count. Now, this is a normal karyotype for a human male. As you can see, the two sex chromosomes, there is an X and a Y. And then all the other chromosomes are neatly paired with a homologous chromosome, and everything is just exactly as it should be. Now, usually when you get these abnormal chromosomes, when you get like one too many or one too few, the resulting embryo is going to be inviable and it's going to result in a miscarriage. However, there are examples, there are syndromes that can result from non-fatal nondisjunctions, such as trisomy 21, also known as Down syndrome. So trisomy 21, as we can see in this human example here from this karyotype, notice that the chromosome that's kind of boxed there is chromosome 21. There should, in a normal sample, only be 2 copies of chromosome 21. Humans are diploid organisms. There should only be 2 copies of each chromosome. But in this example, there are 3 copies of this chromosome 21. Hence the name trisomy 21, tri 3. There are 3 copies of chromosome 21, which that does result in what we know as Down syndrome. This particular chromosome disorder is not fatal. However, it does result in symptoms such as the heart and respiratory problems and varying levels of mental development problems. We're not going to go into it much further. It's just a very common example of a non-fatal nondisjunction disorder. So what about with the sex chromosomes? What if there's a nondisjunction that affects the sex chromosomes? Well, in general, abnormal numbers of sex chromosomes are not going to be as deadly as abnormal numbers of those autosomes. And this is because, one, the Y chromosome is relatively small with very few genes carried on it. And most, at least mammalian cells, we are mammals, and most entry-level bio textbooks that you're going to see are going to follow human examples because that's what's most relatable. In general, if you're just taking a very basic entry-level bio class, you're going to focus on the human examples. And we're mammals, so most mammalian cells can operate with just one functioning X chromosome, or else human males wouldn't be able to function properly because they only got one X chromosome. So mammalian cells can function with just that one X chromosome. So below is a table of a few common syndromes from differences in that whole nondisjunction possibilities resulting in abnormal numbers of sex chromosomes. We're not really going to go through it, but it is an interesting thing to have. And just note that if the nondisjunction occurs in a sex chromosome, it's not necessarily going to be lethal, and it's not always lethal in the autosomes. However, it is more likely to be lethal in an autosome than it is in a sex chromosome. We've been talking a lot about how nondisjunctions are bad and how they spell doom or result in these syndromes that can affect an organism for the entirety of its life, like trisomy 21. But the thing is, not all nondisjunctions are going to be a bad thing. In fact, nondisjunctions can result in new species. The new species that does result from a nondisjunction will have what is known as polyploid somatic cells. So they're going to have more than 2 copies of chromosomes. This is more common in plant species. Some common examples are going to be things like wheat, potatoes, and cotton. So specifically in flowering plants that can fertilize themselves. This is a phenomenon that happens more. It's not necessarily uncommon to find like triploid plants, but it's not necessarily the norm for all organisms. And while it is rare, it can happen that a nondisjunction will result in a brand new species of animal as well. An example of that is the gray tree frog here. We're not really going to get much more into this particular phenomena of nondisjunctions forming new species just because it is still kind of a newer field to study, and honestly, we have more questions than we have answers for this, so let's move on. So there can also be problems with the chromosomes themselves. We're going to get more into these particular slides in chapter 10. I actually think it's really weird that this textbook brought in this particular set of things because this technically has more to do with mutations than nondisjunctions and problems during cell division, but the textbook mentions it here, so I'm going to mention it here, but we're just kind of going to speed through these slides and we will get more into everything that we're going to talk about in the next couple of minutes when we get into chapter 10 and actually talk about evolution and mutations. So the first thing we're going to talk about that is a chromosome problem is something known as a deletion. Now, a deletion is exactly what it sounds like. A segment of that chromosome has somehow been removed. So a part of that genetic code, no longer there. And the next thing is a duplication. Duplications occur when a segment of a chromosome is copied and then stuck right back into that chromosome. So you get that little pink part you can see in this image ends up, instead of just being once like it should be, ends up twice in a row. 2 of the exact same code, one right after the other. It's duplicated. So you can also get what's known as an inversion. An inversion is when you get like a segment of code and then just flip-flop it. So in this example, you have the pink and the green section that just kind of flip-flop and turn into green then pink. So an inversion, you literally just kind of turn the thing on its head. It's in the opposite order that it was before. And then the other thing we're going to talk a little bit about is the reciprocal translocation, where you get segments from 2 non-homologous chromosomes literally swapping places. So you get this code that's not supposed to be on one chromosome completely taken off of its home chromosome and moved to that brand new chromosome. So here we see a big portion of this red chromosome being swapped with the non-homologous blue chromosome. So these parts of the genetic code, since they're on non-homologous chromosomes, don't even necessarily code for similar things. So this is an issue as well. But that's all we really have for you. That's all that we're going to talk about in chapter 8. The big takeaways from this section of this particular video is going to be the first part where we're talking about nondisjunctions and the things that can cause them, where nondisjunctions happen, and the differences between them occurring during meiosis 1 and meiosis 2. Also remember, trisomy 21 is Down syndrome, and it's from having an extra copy of that chromosome 21. Coming from taking many different bio classes where we've talked about this stuff, if you're studying for an exam, they always ask for an example of a non-disjunction in humans, and 90% of the time they want you to remember trisomy 21, and they also want you to be able to recognize it as Down syndrome, because biologists love examples. It's easier to learn things if you have examples. So I hope you have a wonderful rest of your day, afternoon, evening, what have you, and I will see you all in the next one.
Thank you again for watching this video. Don't forget to like, comment, subscribe, and ring that bell if you would like to see more. And if you'd like to follow me on any of my other social medias, the links are down in the description below. Don't forget to check out thereptilegoth.com for all of my articles and blog posts. If you found any value in this video and you would like to help support the channel, please check out my Patreon page. That link is also in the description down below. And a special thanks goes out to my Diamond Dragon patron, Diane B. What you're doing is really helping me fund a dream here. I will see you guys all in the next one.